Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. Good evening, Pa. Good evening, my dear students.
Good evening, sir.
Good evening, Ajay.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much. Okay. Good evening, my dear students. So you are all are warm welcome. Good evening, Kana. So you are all are warm welcome to the another session of the biological classes.
Good evening, sir.
Good evening, Pa. Last class, what we have discussed, what is meant by pleiady, what is meant by polyploidy, what are the types of pleiady, how they are classified according to their chromosomal number, and that things also already we discussed in the previous classes. Okay, so you know very well about the two types of pleiady is there, that is called as what is that, autoploidy and alloploidy. Okay, so before that, when according to their number of chromosomes, for example, suppose the only one chromosome present in it, it is denoted as a haploid. Suppose only one is there. Okay, it may be vary, depends upon their structure. The arms will be vary. It connected with the which one in the centromere. There is a portion or connective link is called as centromere. Already you know about it. Okay. Okay. This is the first one, the haploid in nature, that is denoted as a single N only. Sometimes they may be expressed in small n, sometimes it may be a capital N. Okay, N denoted as number of chromosomes. According to the number, they are divided into so many types. See that haploid, the number is 1. So, N nature. Diploid means each chromosome or a pair itself. Together like this. Okay, 2. Here also 2. Here also 2. So, this is considered as a diploid stage. This is considered as a diploid stage. When you are going to study the, when we were studied in mitosis, meiosis, diploid nature is the diploid nature divided into haploid. Sometimes it happens triploid and tetraploid and hexaploid when you know the one plant genes also addition with the another genes. Okay, so that condition is called as what is that, you know, polyploidy. That condition is called as polyploidy. So here triploid. Triploid means 3 numbers, 3 chromosome present in it. Tetraploidy, 4 numbers, and hexaploidy, 6 numbers. Likewise, we can classify it according to their number of chromosomes that can be mentioned in the form of N. Okay, this is the main things. Okay, see that. The 2 things is that there are 2 major types of polyploidy. Polyploidy means more than one set of chromosome present in a single gene is called as polyploidy. They divided according to their what is that? Nature. One is called as autopolyploidy and allopolyploidy. Autopolyploidy means what is that meaning? In which all chromosomes set or form a single species. Take an example, say a lion, tiger, whatever it may be, any one of the single species you can take, and then there may be what is there is a changes occurs in the chromosomal aberration due to mutation or something else, whatever it may be. Such type of ploidy is called as autopolyploidy. Allopolyploidy. What is the meaning of allopolyploidy? Take an example in this category: tomato and potato. Both are comes under the heading. Which family, my dear students? Potato as well as a tomato. Both are comes under the family. I'm asking. Anybody else? Ajay, Ritika, and Dhanalakshmi. Will you please tell me which family belongs to? Any idea? No idea. Please answer me, my dear students.
No, sir.
Okay. Okay, anyhow, what about Fabaceae or Solanaceae or Malvaceae or monocot? That is called as, what is that? Monocotyledon. You know what about the Solanum tuberosum? Potato is considered in Solanum tuberosum. And Solanum melangina, Solanum capsicum, Solanum darwum. So all these 2 category, potato as well as the tomato, both are considered, the heading is known as Solanaceae family. Okay, Solanaceae family. So they are all considered as single species only, because Solanaceae is a single species. Allopolyploidy. What is the meaning of allopolyploidy? Means different species there. See that in which the chromosomes set off from 2 or more species. Okay, so already told you Solanaceae is one of the family. They took which one, you know, for an example, so for the hybridization process, tomato as well as the potato. Tomato already, you know, it is a fruit, isn't it? Potato, it is the underground stem modifications. When we hybrid with each other, what will be the product? That condition is called as pomoto. That condition is called as pomoto. It is a new species. We can get the fruits in the upper part, and we can get the another one stem root modification is also there in the underground. So this is the new variety, isn't it? Normal variety, you know about that upper part only is there is a fruit in different plant. But at the same time, another one is also. But here, the single species they told, isn't it? Suppose if it is opposite or different, where and a variety of species or more species, that is called as allopolyploidy. See, then example, already we discussed these things is also. What is by autopolyploidy, how their nature, what are the examples already we discussed in the previous classes. What is by autopolyploidy? The autopolyploidy happens due to the accidents of meiosis or mitosis. It is naturally occurring during the cell division process. The time what happened, the 2N embryo also, sometimes it may be produced 3N embryo, 4N embryo, like that. It is naturally occurring due to the cell division process. It is what happened, the divisions also takes place. Where it will take place, see the cell division process, the prophase 1 of meiosis. See that? Because all the chromosome sets of autopolyploidy or forms the same species, they are homologous and attempt to align in prophase 1 of meiosis. Autopolyploidy takes place in the prophase 1 of meiosis, which usually result in the sterility. This usually result in the sterility. What is the meaning of sterility? Sterility means it cannot be able to fuse with the egg of sperm, like that. Okay. Suppose autopolyploidy can arise through the non-disjunction in mitosis or meiosis. What are the example has given here? Many angiosperms or autopolyploidy. Example, grapes, banana, sugar beet. Already we discussed these are the things. Yesterday we discussed. Some of them are plant example has given here. Some of them are plant example. Some of them are animals. Enothera lamarckiana, chrysanthemum. So, okay, these are all what is Enothera lamarckiana. What is the meaning of Enothera? Is a plant or animals? Imagine, mostly plants. This also happen. Okay. Go to the next one is called as allopolyploidy. This is very, very important, my dear students. So many times they were asked the question in NEET. Okay. Allopolyploidy arises from hybridization between two species. Take an example, tiger as well as the lion. Okay. A donkey as well as the, what is that? Horse. Any other plants or animals. Okay. In different species there. Okay. So resulting, the polyploidy carries a chromosome sets derived from the two or more species. Example, wheat, American cotton, Nicotiana tabacum. Tobacco, a botanical name is called as Nicotiana tabacum. Two recently produced allopolyploidy are, note it down, my dear student. This is very, very important. Two recently produced allopolyploidy or, see here, triticale. So triticale. So here, what is it? When you are traveling, my dear students, just listen. When you are traveling, okay, in the bus anywhere in Coimbatore or Bangalore or whatever it may be, Chennai or something else. In between there is a hotel, is there, isn't it? What is the name of that hotel? Will you please? On the way to Kodaikanal or whatever it may be, any one of the bypass, there is a hotel, is there? Okay. When you are stopping that one, they wrote it on their board. This one is called as, what is that? Motel. Motel. Hotel and motel. When you are moving, that time you are stopping at some places for the rest. That condition, that name is called as, what is that? Not hotel. It is a motel. Likewise, motel means motor and hotel, the last letter as well as motor mobility. The first three letter as well as the last three letter is taken from the different, what is the name. Likewise, the allopolyploidy also taken. Triticale. It's the first human mad cyril. Note it down, my dear students. First human mad cyril. Crossed between Triticum durum. Triticum. From here, the triti they have taken. It's also one of the wheat. And also scale cyrillae is the ray. So here the scale is the last three letter is that. Okay. CALE has have taken. So triticale, they have given as a new name for the new plant after the hybridization of these two. These grass between Triticum reverum, it's one of the wheat, and also Cecale cyril is the another type. It's also rye. These two are crossed as a result of formation of F1. Which one we will get it? The triticale of name of this one, of name of this one. This is the first human mad cyril. Okay, note it down, my dear. This is very, very important. Likewise, another one is also, see here, Raphonobrassica by, see here, Georgi. Okay, Corpico Kenko. Okay, the name of the scientist has given here. Cross between the Raphanus sativa, radis, and also Brassica. Brassica is nothing but the cabbage. Brassica oleracea, Brassica protidis. When you are studying the trinomial nomenclature, when you are going to study the trinomial nomenclature, what is the meaning of trinomial nomenclature? Anybody else? Binomial already you know. What is by trinomial? Binomial is nothing but any one of the plant.
Words will be there.
Yes, two names or two words we are calling, isn't it? That condition is called as what is that? Binomial. Suppose we are calling trinomial means, what is a binomial? What are the part is that whether it's a genus name or species name, isn't it? First name is genus name, second name is species name. That is binomial, isn't it? Suppose trinomial means addition of one is that. What is the additional one? Anybody else? Genus name, species name, and
Sir, is it family name?
Badandra?
The family.
No, not family. It has another one. See, last two hierarchy only, classification hierarchy only have to take. Genus name, species name, already know, isn't it? So addition one is what is that? Genus name, species name, and subspecies name is also added. That's why they have given us a trinomial, or hence they are called as a trinomial. What are the names? Binomial means two only. Genus name, species name. Okay. But trinomial means three name we are calling. Okay. We will give three words or three names for the particular plant is called as trinomial nomenclature. Okay. So trinomial means first name is called genus name, second name is called as species name, third name is called as subspecies name. They have given as an example the Brassica oleracea and Brassica and also oleracea, same genus name and species name, but the subspecies will be differ in trinomial nomenclature. Just to go through it after finishing this class or after the 8 o'clock, okay, and above, you just to go and check it. Trinomial nomenclature, who discovered, what are the example, note it down. This is very, very important one. Brassica. So Brassica is nothing but the cabbage. Suppose the Brassica, the Raphanus, both are, what is that? Cross between, as a result, the new one is obtained, that is called as Raphonobrassica. The first name is called as Raphanus. They have given as a genus name. And second one is Brassica is a species name. Okay. Likewise, other examples also given. There are so many examples. They were discovered. They are now also the research also going on. Different plants crossed together as a result. What happened? Other examples also, tobacco and cotton and some other things also. It is also plant related one because we are developing in the agricultural aspect. Okay, and research oriented. Which one is a new one? So for example, Chinese people take an example, say a small pumpkin is there in our area, and there the pumpkin is total 30 kg, 40 kg like that. Even take an examples of sugar cane. They hide the elephants. Even rice also. In Thailand, rice is also what happened in it. Easily hide the elephants. Okay, in our area, but maximum our what is the knee level only, isn't it? Likewise, there are so many varieties have been reported and also produced. Even taken examples of what is that? Animals also. Okay, pig and cow, buffalo. That things also, new varieties also discovered due to this pleiady. Okay, anyhow. Raphna brassica, see here what they have did, the Giorgio and Carpico Henco created polyploidy experimentally in 1920s. Cabbage and radish are agriculturally important plants. Okay, he wanted to produce a plant that had cabbage leaves and radish roots so that no part of the plant would go to waste. That's the things already I told you. Potato and tomato, both are edible part, isn't it? The upper part of the, which one, potato. And tomato is the upper part. And potato is the underground stem molecules. Both are useful one. We need both the character in the single plant. We need both the character. Upper part is also used and also lower part is also used. Okay. So we have to cross in between these two. What happened? We will get it the pomoto. Already I told you. Likewise, what they did, the Georgie. So also did the radish and cabbage. As a result, what happened? See here, he also new species also invented. Okay, that is very, very important here. Okay, so the what will be the chromosomal number? You should note it down. This is very, very important. Sometimes it may be positive result, sometimes it may be negative result. Kenko was able to successfully cross them, producing a hybrid with 2N stage 18. Unfortunately, the hybrid was sterile. Note it down, my dear students. Sometimes it happens. What happened, new result will give. Sometimes it may be a positive result. Sometimes it may be a negative result is formed. And one more example I will explain here. Take an example of sugarcane. Okay. Some scientists, they think sugarcane as well as the bamboo. Bamboo, the size will be very large in nature. Size as well as the width of the stem is also very large in nature. They thought after crossing, okay, the sugarcane will be look like a bamboo. But what happened? The bamboo look like a what is that? Sugarcane. That is also a negative result. Okay. So likewise, they discover. These are the different types of the wheat: wild emmer and domesticated emmer, durum wheat. This one is taken by the scientists. Common wheat. So these are the what is that things. So these are the corresponding what is that things. So what is the next one? See here. The ray Sequels reels, already we discussed it. Triticum ascitivum. Both are having a chromosomes. Cross in between these two, you will get it. The haploid gamete is 7. Suppose if it is double, it is 14. The ray is how many chromosomes present in 14 chromosome is there? That is also a diploid nature. In sexual chromosome, how many chromosomes is there? 7 is there. Okay, but just like a 46 divided into 23 in human being, like that. Likewise, the bread weed also, Triticum ascitivum. Okay, the hexaploidy. Hexaploidy means, what does that mean? Hexaploidy. Hex means 6 into so. Okay, triploid gamete is, see here the 7 is n, isn't it? 7. You have to multiply into 7 into 6. 6 into 7, whatever it may be, you will get it 42. Triploid gamete, how many is that? 3n. 3 into 7, you will get 21. Okay. See here what happened in the cross. So when you are crossing these two, what happened? The 42 as well as the 14. See here, the ray n is 7, wheat 3n is 21. So total, the critical, how many things is we will get? 4n stage. That is tetraploid nature. We will get it 28. This is the normal first one. Okay, 4n stage only. When you are introducing the colchicine, already available, colchicine is the one of the chemical substances that is used mostly for the polyploidy and improve the quality. Okay, likewise. So this is the normal process. Okay, when you are crossing these two, you will get it with how many is there? The 3n here 1 is here. Here n is 7, here 3n is 21. The plant is vary. Okay, this is the ray, this one is a bread wheat. Both are crossed together. The chromosome number is also 7 and 21. You will get it. The new one is called as 1 plus 3, 1, 4n, 7 plus 21, 28. This one, after the mutated by some chemical substances, they will get it the triticale in 8n stage. 8n means you have to multiply 8 into 7, you will get 56. This is the another category. It's advanced, what is that? Multiplications. Likewise, imagine. Likewise, ray scale cereals, diploid sporophyte 14 already you know about, 7 is also there. Durum wheat, triticum turgidum. Here is a changes is there. So tetraploidy is also 28 stage. Both are same. So we'll get it 7 plus 14, 21. After the colchicine introduction, what will happen? You know, it may be multiplied into double. Here is the 4n, here the 8n is double. That is the reasons. So the yield will be more. That is the things. You have to take from here. When you are introducing the plant as well as the plant, the cross between the two plant or two species, you will get the new one. It is a normal one. It will not sufficient for the what is that, human populations. Suppose we need more food, okay, we have to produce more number of what is that, rice or wheat or whatever it may be, like that in acre. So that time what they did, you know, the scientists, they take an example, say MS Swaminathan, Sonara Sarpadi. Okay, it's one of the wheat trees also discovered by the MS Swaminathan. With the help of the colchicines, the colchicine is a chemical substance. So what happened? No, it may be double in that in the productions at the end of the harvestings. Likewise, what happened? You know, they did that condition is called as polypodium. As a result, you will get more number of, for example, per acre, you will get it 100 bunch like that, you know, bag like that, you know. So suppose we are doing this one after the polyploidy interacts, a new variety will get it 200 or 500 like that per acre. So which one is the beneficial one? The 500 will be a beneficial one as well as the commercial oriented. So that is the important things here. Just note it down. This is my dear important. See here, another ones. Rapness. So 2n stage is 18. Diploid stage is 18. Parent of Brassica. These are the two things is that. Okay. Rapness Brassica is a variety of both the characters present. See here, the gamete is 9. Here the gamete is 9. So both are combined together, form as 18. Okay. 18 means what is that? The both the character present. This one is called as F1. After the another things, the fertility amphiploid diploid process. This one is called as sterile conditions. The F1 is sterile. First generation is sterile condition. It may be converted into fertile conditions. Then only it will be survived. Without seed, we cannot be able to produce any plant or animal, isn't it? Take an example. Dolly. Ian Wilmot also discovered the new variety of goat from the udder of the female or male. Take a single, but in the process is called as cloning methodology. Okay. What happened? The single surrogate mother getting the single cell. The single cell produce what is that? Entire animals. It is also called as totipotent in plant as well as already in our about. That is different. But here, what they did know, both the plants are crossed together. The first hybrid will be sterile in conditions. Sterile means what is that? What is the meaning of that one? We cannot be able to cultivate again. It will not. What is that? Planted again for the further use. That is not useful for the human being as well as the population process. So we have to modify it into sterile into fertile conditions. Then only it will be useful one. Okay. Likewise, that's why they were used colchicine. The chemical substance they were used for the colchicine. What happened? You know, sterile condition converted into fertile condition. After that, they may be planted. They may be advised to the what is a farmer. Okay. So this is very, very important. These are all done by which one, you know, ploidy. Okay, see here. So examples of polyploidy crop plant. Note it down. Sometimes they may ask the chromosome numbers in this part. What are the ploidy? You have to multiply in the 4n means what is that? You have to calculate. Potato, it is also all are autopolyploidy. No, see here that two sweet potato autopolyploidy. Remaining are allopolyploidy. Potato autopolyploidy, same species. Banana autopolyploidy. Beanut autopolyploidy. Sweet potato autopolyploidy. Okay, these are all our same species. See that 4n, 3n, 4n, 6n. So this is ploidy nature, 4n. Okay. So we'll get it. What is that? 48. 4 into n, 48. What is that? 7 into 7 into 7 into 7. Or like how many things is there? Divided by 4, you will get the single n. 3n, see here, 33. So the 11 is there. Banana, the chromosome number is 11. 4n here, see here, peanut. So just imagine this is 40 means 4n is equal to 40 means what is the meaning of here? 10 is a single one. Sweet potato 6n, see here, 90. So 15 is here. Tobacco, allopolyploidy. Cotton is also allopolyploidy. And wheat is also allopolyploidy. Oat, allopolyploidy. Sugar cane, allopolyploidy. Strawberry, allopolyploidy. They have given here 6 and 8. 4 is there. So total 10 has given. The 4 is the autopolyploidy. Remaining 6 is the allopolyploidy. See here, tobacco 4n. So we'll get it 48. Cotton 4n, 52. Wheat 6n, 42. Oats, allopolyploidy 6n, 42. 4n, 4n, 6n, 6n, 8n, 8n. Like that you have to calculate and corresponding chromosome number also. Maximum, these portion maximum they were asked. Potato, banana, and tobacco, cotton, wheat, and sugar cane. Sometimes rarely asked the strawberry also. You have to practice it. Okay. See here. So this is another one. These are the things is that due to the polyploidy, what happened? Due to the chromosomal nature is demolished or some changes occurs within the chromosomal numbers. What will happen? It will also lead to some diseases. That condition is called as what is it? Chromosomal abnormalities. Normalities already available. Suppose if it is due to the addition or deletion or substitution, transition, inversion. Likewise, there are so many changes is there. Okay. As a result, what happened? The chromosomal number also change, whether it may be less or gain. Okay. It may express into the generation, means it's lead to some diseases or syndrome. Okay. See here, what are the symptom and abnormalities in the things? They have been, what is that? Tabulated here, chart. Likewise, see here, the chromosomal abnormalities. The structural abnormalities. Suppose if it is the chromosomal is the chromosome contain 300 chromosome or 100. For example, 10 chromosome alleles is present in it. Suppose a single alleles, a single chromosome gene is also altered. What are the alteration takes place according to the structural aspects? Deletion, ring chromosome, duplication, translocation, insertion, isochromosome, inversion. These are the changes occurs in the structural aspects. What is the meaning of structural aspect? The nucleotide will be changed. What happened? Automatically that expressed itself in the next generation. Suppose the parent has 10 chromosomes like that. Imagine. Often it has 9 chromosomes. There is one chromosome is lack or one nucleotide is lack. So it is a syndrome. It has some specific effect on his body or something else. Okay. These are the structural. Autosomal abnormalities. What is my autosomal abnormalities? There are 22 pairs there. Last one pair is sexual chromosome, isn't it? So that is the given. See, Cray-Due-Chart syndrome, Down syndrome, Padu's syndrome, and Edwards syndrome, and Philadelphia chromosome. These are all autosomal abnormalities. Note it down, my dear. Autosomal means only for the somatic cells. And sex chromosome means XY related. Don't forget, my dears. Sex chromosome abnormalities mostly difficult in nature. Turner syndrome, Klinefelter syndrome, triple X syndrome. Note it down, my dear. XYY syndrome, and 40XXX male, 40XXY female, and see here 46XY female. Normally XY is male only. Here the opposite manner it gets given. You can see XX male only. XX maximum female is denoted. But in this category, XX is a male. But here XY is a female. And also testicular feminization syndrome. These are all the different. Fragile X syndrome and intersex. These are all sexual chromosomal changes or abnormalities takes place due to the physical and chemical and biological aspects. So these are the things that have been reported. So first one is structural aspect, Second one is autosomal or somatic cells. And third one is chromosomal, sex chromosomal, is XY related. Okay, see one by one very deeply. See here, already we studied in the 12th standard. So this is the chromosome structure. See here, this is the chromosome structure, normal one. Suppose the portion of the AB is removed in this portion. Okay, removed. That is called as terminal deletion. This part is completely removed, means this will be the effect of the next generation, isn't it? It is removed. After that, it may be inherited to the next generation. As a result, what happened, it will not come into the next generation, isn't it? So this is called as terminal deletion. This one is terminal deletion. Intercalary deletion. See here, intercalary deletion. What is present here? EF is only present here. Okay. Remaining completely absent. So the upper part as well as the lower part. See here, this example, take an example. The EF only present in this category. And completely removal of this portion. A, B, C, D, G, and H. Okay. The EF is center portion. Okay. You compare with this only. Okay. This is the normal one. You have to compare these two. Okay. This one is a normal. The terminal means this portion is completely removed. The first one. Second one is what is an intercalary. Intercalary means in between. Deletion. Deletion means what is the meaning? This portion is completely removed. Which portion? The EF is completely removed. So this is the chromosome structures. It can express the next gen. So a removal of this portion, what happens? It affects also. It affects some enzymes or hormones, whatever it may be, like that. Okay. And duplication. What is see here? Duplication. See the last two digit. GH is also again duplicated once again. Okay. It is also gives some effect in the chromosomal aspect. And another one is the inversion. Listen here. A, B, C, D, E, and F, and G, and H. Here, see here. A, B, C, D is there. Here, the GH is upper changed and invertedly present. EF is down and GH is upper portion in this category. See that in this portion. GF is here is there, but in this portion, what happened in this chromosome? You can see the upper part. It will be changed. That is called as inversely changed. And heterozygous translocation also. Heterozygous translocation. Heterozygous during the crossing over already over. But this is the normal chromosome. This is also normal chromosome. In between, what happened? The interchange of the chromosome from the here to here, and here the portion is also transferred to here. Okay. In this category, I will show you a small one due to the whiteboard. Then only it will be better for your things. See here. See here. Take an example. This is the one chromosome, isn't it? Take an example. So this is the one chromosome. This is considered as A. Okay. Then next one is called as what is that? You consider as a B. Okay. This one is another chromosome. So this is considered as a B. Okay. When it is a common contact closure during the time of what is that meiosis, what is the crossing over takes place in this portion only. Okay, this portion only. What happened in this portion? See here, the same diagram. Okay, the same. What is that? Same diagram. See that the first portion, what will happen? So you will get that. This is the one part. So another part is also there. See here, there's another part. Here the crossing over takes place, indeed. So this part completely move to in this portion. So in this part move to in here. Okay, you have to change it. Okay, so then only you will get the different structures. See here. So this portion is different. So next one, the orange color already in over. This is another chromosomes. Okay. See here. This is the portion. This portion is the remaining portion is here. See this one. This remaining portion is here. Here interchanges also takes place, isn't it? So this is the things. Okay, see that one. Okay, once again I have to move on to that one. See here, this one. Okay, see here, this is the portion. See here, this is the white portion is the similar. These are the sister chromatin. Here, what happened? No, this is also same, isn't it? You look like this one, same like this. Okay, here the changes is there. Here, the a particular portion is also crossed with here or joined with it here. Here, the portion is also joined with the another chromosome. So as a result, the crossing over also takes place. Okay, so we'll get that different. That's why it has a heterozygous translocation. Homozygous means what is the meaning? Same chromosomes, they can change themselves. Okay, this is the homozygous. As a result, what happened? It will get the new one. Okay, okay. Go to the next one. So duplications. So duplicated gene. See here, this is the one part is already given here. The gene is again what is that? Duplicated. As a result, what happened? No, the two set of chromosomes, two types of genes also present. Same gene, single gene is here. Here, the two genes are present. As a result, what happened? The chromosomal structure also changed. So it will give some different effects. These things is, suppose take an examples. This is the color blindness like that, imagine. This color is also a color blindness. Imagine means it may be high. So both double time it increases, double time. Duplicated genes also present in a single chromosome. It gives a specific effect. Okay, this is the duplication process. Already we discussed. This one, see here, the chromosome is the normal one. Balanced translocation. Translocation means the portion is, it will be rejoined. Okay, this portion, the partial portion is also joined here. This portion is also joined here. As a result, also what happened? It came the changes. So the same thing, deletion. The particular part will completely remove and join together. This condition is called deletion. Duplicate. This portion is also duplicated. And what is inversion? Inversion means reversibly. Already I told you, GF, that things. So insertion. Translocation. Translocation means what does that mean? The particular nucleotide also changed the 4th place to 10th place. So as a result, some change also occurs. See here, these are the changes. Chromosome 4, chromosome 20, they told. Okay, translocation is also similar. This is the another. First one is deletion, duplication, inversion, and insertion. Insertion additions. Okay. Here, translocation means the place will be replaced in somewhere else. Whether it may be a 4th place. Suppose the chromosome present in it may be placed in the 10th place or 20th place or 48 or 106 like that. This is the symbol diagram. Okay, got it. So as a result, what happened? There is a changes occurs in the chromosome structure. So it will express itself in different aspects. Okay, okay. Then we will pass on to the next one is syndromes. Syndromes. Chromosomal abnormalities. What will be the changes within which occurs within the chromosome? What will be the karyotypes? Okay, this is very, very important, my dear students. Don't forget this part. Down syndrome. It comes under the heading is known as trisomy. Note it down, my dear. Trisomy 21 type, translocation type, and mosaic type. These are the 3 types that have been reported in this Down syndrome. Most of the cases they were asked which one is the first one only. See here, 47, XX plus 21. Okay. So here 23 chromosome is there, isn't it? 21 plus 22, 23 only. But sometimes what happens now, 47 will obtain. That is called as Down syndrome. It is also one of the trisomy. Translocation types. See here, 46, normal one, XX, this is the 48. Finish it. But here minus 14 gas given and plus T. These are all complicated only. No need this type. Sometimes it may be very rare cases only. Trisomic is the first one. Mosaic type sometimes they are asked. 46 XX, 47 XX plus 21. Turner syndrome, monosomy. We'll discuss how it will be happen. And Turner syndrome is also another type of syndrome due to the chromosomal abnormality. This is also considered as monosomy. Note it down, my dear students. Turner monosomic X 45, X. Klinefelter syndrome. Note it down, my dear. This is also very, very important. 46, XXY. See here, the female has the sterile condition. This condition is called sterility. Okay. Suppose, see that XX or XY. XY is also another type. Mosaic type is also the XXY. This is also a XXY is also mosaic type. Already we discussed. Klinefelter syndrome is also mosaic syndrome. Edwards syndrome, trisomic, 18 types, 47XX. Okay, 47XX, what is the meaning? 48. Okay, 48 divided 2 means we will get 18 plus XX. Mosaic type, see here, there is a type. One is reduced from the previous one, natural one. Purdue syndrome, trisomic, 13 types, translocation type, mosaic type is also there. Trisomic and also 9 mosaicism, trisomic 9. Okay, 47XY plus 9 here, syndrome. Cry-Due-Chat syndrome, deletion. These are all deletion type, fifth and position of the 15th. And Wolff-Hirschhorn deletion is also deletion of the 4th, P and 14th. Syndrome, trisomic 8. Okay, trisomy 8 is also 47 into XX plus 8. Mosaic deletion. These are all other different types of syndrome have been identified by the scientists and also tabulated here in the form of, and also their chromosomal abnormalities, how it happens, and also what are the carrier types. That things also given here. See that one by one we will discuss it clearly. This is the normal one. Normal male. This is the normal female. So normal male means they have the X chromosome as well as the Y chromosome, isn't it? Suppose the Klinefelter, okay, syndrome means what is the, see that XY is the addition of X. What is the meaning this one? The male have the female character extra. Okay, this is called as Klinefelter syndrome. My dear students, this portion they ask the many times. XY already you know, but the male character having the female extra. Okay, it's a third generation they told, isn't it? This portion. See here, XYY. What is the meaning of XYY? See that XY is the male, but they are having the Y is another Y chromosome is also present, and it is also one of the abnormalities and also syndrome. Normal means 46 chromosome must present in each and every human being. It's a normal one. Suppose any one of the lack or addition, that condition is called as ploidy. Already we discussed in the introduction point. Okay, here what happened, XY is the normal one. The addition of Y chromosome is that. This is the addition one. So here what happened? Plus 1. Here also see here, the Y chromosome rearrangement is there. Here what happened? Deletion of Y. Q like that they told. This is the normal one. The deletion, this chromosome is completely absent like that, These are all are male related one. The same one is also mentioned in the female. See here, normal female is that this one is what is XX is the normal female. Turner syndrome. Turner syndrome means, you note it down, my dear students. Here the X, here X is deleted by the XY. X, this is also XQ, X more. These are the Turner syndrome. Trisomic X syndrome. X means, see here, all the three are X. What is the meaning? Feminine characters will be very high in nature in this category. So these are the another things also given. Okay. See, disorders and description and associated cardiac abnormalities. Okay, already we discussed this point also. Okay. So, and trisomic Down syndrome, trisomic 18 Edwards syndrome, trisomic 13 Purdue syndrome. These are all are what is a trisomic nature. Monosomy is also sometimes is there. That is called as Turner syndrome. Single X chromosome in females only. Only one X chromosome is present in it. That condition is called as monosomy. That is also another things. Okay. Likewise, the chart has given here. Go through it in your book. You will get it the idea. And next one is pedigree analysis. What is the meaning of pedigree analysis? Already we discussed. A record of inheritance of certain traits for 2 or more generations present in the form of a diagram of family tree is called as pedigree. Family tree, note it down, my dear students. I already told you. A family tree or pedigree is drawn for families having genetically transmitted diseases or trials. Suppose any one of the family and grandfather or father or grandfather, he has or she has some specific diseases. How will you identify it? How will you identify it? With the help of the pedigree analysis only. Suppose the parent, grandparent has the disease, have the disease. How will you identify it? With the help of the pedigree analysis. The family tree or pedigree is drawn for families having genetically transmitted disease or triads. What is the meaning? The family tree or pedigree is drawn for families having genetically transmitted diseases. Okay. Genetically means chromosomal related one, not for the somatic cells. Okay. Cancer is also one of the somatic divisions, isn't it? It will not spread through the other person. Okay. See, this is constructed based on the information gathered about all members of the family over many generations. We have to, how will you construct it? You have to take from the 4-5 generations before your father or mother. Then only you will get it the, which one, exact result. Okay. So that is the things. See the pedigree chart. This is male or female. This one is round already in our a female. Okay. See here. The carrier female. When you are putting the divided into two halves with a light color as well as a dark color, this is also what is a carrier of the female. She married a normal woman, normal male. Okay. So as a result, what happened? These are the possibilities there. One is the normal female is possible. Another one is hemophilic male. This is affected one. And the normal male is also possible. These are the three possibilities in what is that? Likewise here, normal girl, and see that this one is called as carrier female. The last one is normal male. Okay. Here what happened, see that after that he married some persons. What will happen here? See, these are the normal. This is, what is that? Carrier female. This is normal. This is normal. This is carrier. Likewise, the pedigree chart we can construct. As a result, we can identify the which person which will be in future also we can find out. Suppose the male or female after you or your generation. Suppose if you have any difficulties like the color blindness, like that, whatever it may, sugar, whatever it may be, we can calculate whether male, suppose father, suppose if you identify the before generation, father or mother or grandfather or grandmother, you have to put the chart and you will get the ideas which one it comes out to next after you. Like that, you can come to the conclusions. This pedigree is represented with the certain standard symbols already we discussed for carrying out of symbol Mendelian analysis involving a recessive or dominant alleles. Certain clues or simple rules are sought from the pedigree. In the case of recessive allele, for example, characteristic conditions can appear in the progeny apparently unaffected parents. Moreover, two affected individuals cannot have unaffected child. So, affected individual cannot have a unaffected child. So, must be affected, like that they told. So, both the parent are affected by particular disease means what happened? The child also must, what happened? Affect. It will not, unaffected child will not get it. That is very, very important. That's why we will, what is that? Construct the pedigree analysis. Okay. So this is very, very important. So already we discussed these things also. The square is also denoted the male. Suppose if it is put 2 is called as 2 male, and proband male, and decased male, affected by history, examined male, and fraternal twins, identical twins, and adapted males. These are the things also comes for female. These are the extra, sex is unknown. Suppose if you are putting like this, the square is in the this portion. Okay, there is unknown like that. Lived 1 day, and stillbirth, it's not birth. Suppose we are putting the plus in the center, you will get the stillbirth. Miscarriage, progeny, this one is called as P. So small dot like structure, and also put P inside it. What happened? That is pregnancy. This is marriage, male as well as female. This consanguineous marriage, this one. This is extramarital mating, divorce or separations, no children. These are all the symptoms that were used for the pedigree analysis for the future and also aspects. Next one is very, very important. We will finish it up to this level only today. Hardy-Weinberg law. This is most important one for the behavior of genetic populations. Note it down, my dear student. This is very, very important. Suppose the chromosomal changes occurs, we do not know how to calculate it. This family is also our relatives. This is also now our relative. Okay, this is maybe our grandfather, like that. Okay, there is a common misconceptions that a characteristics associated with the dominant allele increase in frequency because of its genetic dominance. This misconception is refused by a principle called the Hardy-Weinberg equilibrium. Note it down. Hardy-Weinberg law. Which one is refused? Misconception is refused by the principles called as Hardy-Weinberg law. What he stated, how they calculated. See that in 1908, G.H. Hardy showed that in a population in which random matching occurs and in the absence of the forces that change the proportion of the alleles, the original ratio of dominant alleles to the recessive alleles retain the retardation to the generation. Sorry, generation to generations. What is the meaning this one? Normally, you know, in previous ancient ages, they will not get the correct partnership, isn't it? Randomly matching. So as a result, what happened? Which chromosome, where they segregated, they will not understand. They will not come to the conclusion. So because they will not aware of what is the pedigree analysis. So that time, what happened occurs, and in the absence of force that change in the proportions of the alleles, the original ratio of dominant alleles or to recessive alleles is retained from the generation to generation. So which gene is we need dominant allele, dominant characters we need means we have to put the which one, you know, pedigree analysis, that things is also. So we need the alleles. So we can get the alleles from these equilibrium aspects only. This was constructed by the name of the scientist Hardy-Weinberg. That's why the law is also named as a Hardy-Weinberg law. Okay. It analyzes the behavior of a gene in the population. This is very, very important one, my dear students. Okay. See that. To demonstrate this, they examined the behavior of alleles in an idealized non-evolving population in which 5 conditions hold. Okay. No mutations, isolation from other populations, large population size, and random matching, no natural selection. These are the, what is that? Conditions is also used by the name of the scientist Hardy-Weinberg. Okay. So as a result, you will get the one equation, that is Hardy-Weinberg equation. Already you studied in the max classes. Okay. P plus Q whole squared, that is equal to 1. This is a statement. Okay. So how will you split here? P squared plus 2PQ plus Q squared, that is equal to 1. This is the equation of Hardy-Weinberg law. How it be constructed, we will see one by one in the next classes. Okay. Today, what we discuss, so what are the things, what is my polyploidy, what is my ploidy, alloploidy, autopolyploidy, that things also discussed. What are the examples? Okay. And also, this is very, very important. Triticale, Rapna, Brassica, how they will be, okay, mattered, cross-breeding done by the scientists. As a result, what will be the new species they obtained. Okay. So these are the chromosomes. They ask the questions. Haploid gamete contains, for example, a Sequel cereals contain haploid gamete. How many chromosomes is there? 7. Suppose if it is diploid, 14. Suppose triploid, that is 21. So how it will be? The new one, you will get it there. That is named is called as triticale. These are all comes out of the known as Poaceae family. Okay. When you are introducing the colchicine is the substance, what happened? It may be double of the previous one. So that the things, okay. And then, so these are the another one, Rapnus, Brassica. Okay. These are the 2 plants also. First one, you will get in the sterile condition. After the hybridization, you will get the fertile one. Amphi-diploid natures. Okay. What will be the chromosome? These are all asked in the mathematical question aspects. They may ask the question in the mathematical. Rapnus is 2n stage, diploid stage is containing 18, Another one is also Brassica is also contained 18. When you are hybrids, you will get the F2 generation. What will be the nature like that? So 4n is a 36. Here you had it. Okay, 2n plus 2n, you will get it 36. So 4n is equal to 36 only. Okay, how many percentage? Sometimes they may ask this one. So what are the examples? Already we discussed types of polyploidy. 2 types of polyploidy already we discussed, and also chromosome number is also okay. So these are the 2 things, and structural abnormalities. What are the structural abnormalities also there, and the autosomal abnormalities. What is that? And sex chromosomal abnormalities. Okay, these are the chart. This should note it down, very very important things also. Okay, and then so what are the changes? The terminal deletion, intercalary deletion, duplication, and inversions. How it will be changed? These changes occurs. What happened? Okay, the chromosome is also changed, and also another one is the translocation, changes of the chromosome in the one place to another place. What will happen? It is also lead to some disorder or syndrome in the generation wise. This is also observed by the name of the scientist. Duplication already given in here, and also another one is called as a balanced translocation. The particular part is also changed, removed. Okay, sometimes it may be interchanged. That is called as crossing over. Also takes place during the meiosis likewise. And these are the changes: deletion, duplication, inversion, and insertion, and translocation will be discussed. Okay, very deeply. These are the changes occurs in the chromosomal aspect. As a result, what happened? Already discussed. So these are the chart. How it will be. This is very important. Down syndrome, Turner syndrome, Klinefelter, Edwards syndrome, Porter syndrome. This is very important one. Okay. So here, how many chromosome is that? This is very important. So many times they were asked the question. Just go and refer it. We'll get the ideas. So these are the male as well as the female changes. What happened? This is also important things. Okay. So try something. Okay, my dear students, we will finish it up to this level. Thank you for the participant. Thank you so much. Thank you, ma'am.
Thank you, sir.
Thank you, Ajay. See you.
Thank you, sir.
Thank you, Kana.